Yes. Yeah. Thank you, Akshay, for joining. Um, I think we should probably get started. Yeah. So, dear friends, thank you so much for joining in Sciversation with me. We have two elite guests, very prolific and very terrific scientists, and what more, they are scientist couple, Abhishek Sengupta and Priyanka Narad. In fact, I've known Abhi primarily for little more than 4.5 years, and what we usually do is, to get inspiration, we keep on having these Sciversations. Sciversation is conversation taking up, science with conscience, and we'll just, have some kind of questions extempore after their individual presentations. To start with, I'll quickly introduce Abhi. Both Abhishek and Priyanka have illustrious years of experience, close to 2 decades. While Abhishek, has been working at MIT for the last 16 years, Priyanka worked for MIT University for about 14 years plus, and then she moved to ICMR headquarters, where she's serving as scientist, heading the research division over there. Uh, uh, while both of them have done, PhDs in, integrated multi-omic based approaches. Abhishek has done BSc honors and followed by MSc in UK in Nottingham, I believe, and then he moved back to India, to do his PhD, and the same applies for Priyanka as well. In fact, together they have prolific publications. Some of them largely, in lieu of microbial genomics to multi-omics, AI-driven, bioinformatics, and of course, the most important, the wastewater-based, treatment methodologies, which they've worked on a highly cited, works. So with this little introduction, let's welcome Priyanka and Abhishek very warmly, and I personally thank them, for their time. That too, at a pretty late time. In fact, they have just come from their offices in chilling Delhi cold, and they have been here. Thank you so much, Abhishek and Priyanka. Over to you, please. We have your presentation for 15 minutes, and we'll have Q&A, extempore. Thank you so much.
Okay. Thank you, Prash, for this invitation, and it's our honor to be here in this diversion versation session, and it's been a long time that I'm linked to Prash with different projects and different uh, uh, like no research sessions. So thank you, Prash, for inviting me, and over to you, Priyanka, Dr. Priyanka.
Okay. Thank you so much for the invitation. And it is indeed a pleasure to be joined by students as well as researchers. And let's hope we keep up the topic and also we are able to answer the questions as and when they come. So we'll start with a short presentation that will just be covering.
I'm I'm sharing the presentation.
Yeah. So this will just be covering what we did in the past, maybe 14 years of our research career before I moved to ICMR. And some of the work is also continuing in terms of our research scholars, and it is an ongoing process, and some of it has already been completed. So we'll just quickly take you through what our area is and how do we go about it. Can you go full screen?
Yeah. Yes, please. Yeah. Uh, yes, please. You can make it full screen. Yeah.
Um, I can still not see full screen.
Uh, can you try pressing F5, Priyanka?
It's not at my end, Abhishek. Can you do it? Are you able to put it on full screen?
Just a minute. Is it fine
Yeah.
now?
Yes, yes, yeah,
Yes.
perfect, yeah.
Okay. So I have already been introduced just now by Prash, and I am working in the area of bioinformatics since the last 16 years now, and it has been a great journey. And maybe when we talk more on extempore, we will talk about the journey that was it through. Go to the next slide, please. So, what I started today was to talk about the AI imperative, because what is my new role? It is more like towards administration, and at the headquarters, we are more looking forward towards the AI based approaches. But what is important is that we also talk about trust, why it is important in case of AI that you follow the matrix, and you are also, it is no longer something that is uh, a standalone. It is deeply embedded in healthcare decision making, and however the accuracy alone is no longer sufficient. What truly determines adoption, scalability, and regulatory acceptance today is trust. So I will discuss, briefly touch upon why trust has become a central metric for AI in healthcare, and how structured frameworks are available like Matter, which helps us operationalize trustworthy AI in real world biomedical applications. We've also seen how AI is transforming healthcare from diagnostics and imaging to genomics, clinical trials, and public health. In this transformation, however, the real measure of AI success isn't speed or accuracy, it's all about trust. When a doctor, policymaker, or patient interacts with an AI based system, they must have the confidence in its reliability, transparency, and fairness. So, trust no longer is a concept, it has become a quantifiable performance metric. Next slide. So, here I talk about Matter. So, a trustworthy AI basically relies on five pillars broadly. And Matter is an integrated architecture that standardizes the evaluation of complex AI systems, ensuring they meet rigorous standards for performance and ethics. If I have to break down the acronym, M stands for metrics, which is quantitative performance indicators such as F1 score, robustness, etc. Assessment tools such as softwares and methodologies can be used for assessment, such as LIME, IBM, AIF 360, and there are other benchmarks that are available depending upon what you're working on, what you're looking at to assess in your tool. Then comes trustworthiness. It requires model integrity, security, and ethical alignment. Transparent, where we need clarity regarding data, model behavior, and decision logic. Explainable, which provides human understandable reasons for outputs. And reliable, where the performance consistency remains the same over time and across domain shifts. So, if I have to summarize it, I will talk about reliability, fairness, explainability, safety, and accountability. Reliability meaning it performs consistently, fairness meaning it treats all groups equitably, explainability meaning users understand how it works, safety meaning it does no harm, and accountability meaning someone is responsible for its behavior. These five principles define the trust architecture that we should be trying to operationalize when we are talking about AI based tools and their assessment. Next, we move on to the next slide, please. So this is the framework that I am going to be briefly touching about. So within Matter, the first layer focuses on data quality, and that's handled by the metric framework. which we talked about in the previous slide. Imagine data as the DNA of AI. If the DNA is corrupted or biased, the phenotype or the model's behavior will be flawed. So Metric divides the data quality into 5 clusters and 15 dimensions, covering everything from completeness and representativeness to provenance and accuracy. So this ensures that even before we train an AI, the foundation, that is the data, is trustworthy. So this is a specialized framework which you can see has an inner layer, measurement process, timelines, representativeness, informativeness, and consistency. And then there are outer layers which defines the concept behind these individual process. Like for example, if I'm talking about the measurement process, I will talk about device error, which will talk about accuracy and precision. Then human induced error, which is noise, labels, and which is then completeness, source credibility, where is the data coming from, who is processing it, how, what is the credibility of it, then is it representing a variety of information, is it the depth of data that we're talking about, target class balance, so on and so forth. So all of these are things that you should be taking into consideration when you talk about data, and also when you are starting your framework to build the AI, because if your base is strong, your model will automatically be turned up into a useful outcome as a in the healthcare domain. But if the base or the data is weak, you will never be able to take it up into an operational model which can work across different domains. Next slide. So I will talk about two of our projects in very briefly, where we worked with this data. First one was a DBT funded project where we had worked up on the personalized prediction of cumulative pregnancy outcomes in IVF. So it started with a collaboration with Sir Gangaram Hospital, where we collected data from 2018 to 2020 in the initial first phase, though we've moved up to 2022 as the retrospective data now, and we are trying to build up the final model, which will be able to take into consideration the pre and post treatment parameters. Then data processing was performed on these. These were IVF cycles where there were failure, which there was no clear cause that could be identified. And this leads to a lot of emotional, physical, and financial burden. Existing models are not applicable because of the descriptors. They are models that are available outside India. There are models in UK, US, as well as in Europe, other countries, Edinburgh. There are many models which predict the success rate of IVF, but they are not applicable because the descriptors in India are different. We have different age limits for IVF cycles in India. We have different reasons for why they are not able to process it. Next slide. So what we did here was we collected this data, and we had this architecture where we had put the data from Sir Gangaram Hospital. The original data set had 2020 records and 79 features in total, and the outcome was live birth or negative. And in this particular presentation, we are presenting the pre-treatment model where we had taken into consideration number of IVF cycles. So basically, not getting into the technical details, why we were performing a pre-treatment model first was that when a patient walks in, what is it that is available to them in terms of information, and what is the outcome that they are getting. So this is what we did here.
Uh, Priyanka, Abhishek, am I there?
Studies where what was the Y chromosome microdeletions are a leading genetic cause of male infertility. However, some men with Y chromosome microdeletion have focused spermatogenesis, and sperm may be retrieved in such cases. These men can opt for ART for biological parenthood, and currently there is a lack of objective measure for the success rates of ART, and so for this we built this model which was able to, which was on the basis of locus STS markers and DYS markers, the user details and detected Y chromosome deletion or subdeletion based on locus STS and DYS markers were put into the model, and it was able to predict the clinical pregnancy rate, fertilization rate, and sperm retrieval date in this case, which led to a fertility test report. Now, this work has been transferred to an industry which is already selling a PCR kit for the Y chromosome microdeletion. And along with this kit, when you give this model as an auxiliary to the kit, it is going to work well for the clinicians to find out what the results interpret, because in many cases you have an outcome where you are not able to make a decision. So it is just a clinical decision support system that was built in case of this model, which was built by us, and now it is with the industry. So let's hope it goes to the market soon and it is being able to used by a larger public. So with this, I think I have finished my part of the slides and I just hand it over to Abhishek. Yeah, I have already spoken about it. You can go to the next slide and you can start.
So thank you, Dr. Priyanka. So I'm Dr. Abhishek, and already Prash has spoken about me that I'm working in Amity since long time. It's been almost this coming March. It is going to be uh, 17 years at Amity University Noida campus. So I'm an associate professor there. So me and Dr. Priyanka were the PI, co-PI of the lab, and we started this lab long back, and the name of this lab is Systems Biology and Data Analytics Research Lab, SBDA Research Lab, where we focus basically on transforming the way we study, diagnose, and prognose human diseases, right? Human diseases using computational techniques and methods. So we combine various data, various omics data. So our main focus is omics data, though our first project was dealt with clinical data, the DBT project that we successfully uh, ended with a product, and it is still yet to come to the market as one of the of its first kind in India, with Indian cohort data. So, but mostly our work, the students mostly work on omics data, different genomics, transcriptomics data, and also proteomics and metabolomics. My own PhD was on metabolic networks, network modeling, basically. So we examine disease
Yeah, sorry,
network
Abhishek, are you
utilizing.
moving the slides? I'm sorry. Yeah.
No, no, I'm not. I have not moved
Okay,
yet.
okay, sorry. Yeah, yeah,
I'll just have two slides.
yeah.
So so I'm just telling about what we usually work in our lab. So we use advanced bioinformatics, AI/ML techniques, and basically genome scale metabolic reconstructions. That is GEMS. So that is through GEMS, we try to identify targets. We try to identify. We study the resistance in in various pathogenic organisms. So these are the few areas where we work, and also the disease that I we specially work on is one of the autoimmune disorder, being is vitiligo and its pathogenesis, understanding its complexity, and we have been quite a long time. We have been working on that, and we have there is no database of vitiligo, pathogenesis and its various molecular aspects of it, and ours is one of the first kind again in India. So we are coming up soon with the uh, third version of Vitiligo Information Resource VDB. We call it as VDB. Uh, uh, Soon in 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 the social platform. So with this background, we also utilize machine learning technique, multi-omics, data analytics, and our area is, as you see, as you have seen, that we work on the areas that we work on are reproductive health. Recently we started a project, and we got a project and on mental health. It's a multi-institute project with NIMHANS, AIMS Delhi, and PGI Chandigarh on schizophrenia and cardiometabolic disorders. So it's a methylome based project using AI. So it's a big project. So, and also we work on gut brain axis, gut brain axis in autism spectrum and depression. So also we work on infectious diseases, and basically we work on the uh, uh, on mostly on the genome scale models. We prefer to work on genome scale models, and through genome scale models, how we can solve various problems and come up with solutions. So these are the different works that we have been doing in our lab. So what one of the it's interesting work at the intersection of uh, reproductive health and microbiomics that we and also machine learning that we have worked and published. This was my first PhD student who worked on this data, and this work is how the reproductive or vaginal microbiome, how the dysbiosis uh, uh, in uh, in the vaginal microbiome can lead to uh, uh, pregnancy complications like preterm birth, uh, like uh, and prom, all those different pregnancy complications that it may lead to, right? So this is what what we have done in where we have taken the microbiome data, the 16S rRNA data, and we have processed those data. We have created a pipeline, uh, new pipeline, other than the Mothur or related pipelines, so which is the traditional pipeline which people use. We have used a little bit updated pipeline, and it came to be very successful. In one platform, we have used all the data, and we have done all the uh, alpha diversity, beta diversity, and various statistical analysis, Parmanova analysis, etc. And then we have found out some really good results from that. The really novel results from that. We have identified, other than lactobacillus and Gardnerella, we have identified three, four the uh, new bacterias which are uh, responsible for preterm birth and also preeclampsia. And also the functional pathways and few pathways which have not been uh, reported before, and also few genes. So this was the novelty of our work. So we have taken like almost 3757 vaginal microbiome samples, 16 rNA samples, metagenomic data from 5 public databases, and categorizing them into PTBN, that is preterm birth, and term birth, right? So it is a uh, uh, control based studies, disease control based studies. So uh, we have what we have done, we have classified those samples on the basis of race. So this is one of the very interesting study that we have done, that we have seen that uh, in different races, like ethnicities and races, the how the microbiome or taxonomic diversity varies from one race and ethnicity to the other. And we have very well proven it using AI based tools, and the whole pipeline that we have created. So and also this is cross-sectional study because it is among the races, and also we have done a longitudinal study. So there we have found few new findings. Taxonomic classification and functional profiling we have done using. So we have identified a new tool. I would suggest the students to also try this tool, other than Kaiman, other than Kaiman Mothur, that is parallel meta 3. So we have used parallel meta 3 because it has everything in it, and it is a Linux based uh, tool, and it has like no so that is an amazing tool. So and integrated systems by, so we have used also network based studies, that is systems biology studies. We have created lots and lots of networks and network based approaches and machine learning approaches to find out those key microbes, pathways, and genes associated with preterm birth. So you can see here the ROC curves that we have got uh, from these studies, and also the features, different features that we have identified, feature identification. So this is what I was talking about, our key findings through this work. That is, key microbial biomarkers that we have found out is Shuttleworthia, Megasphaeria, and it has been already cross-checked and validated from the existing literature, that yes, they have role, but it has not been taken into too much. It has not been given too much importance, but through our publication we have proven that these organisms, other than lactobacillus and gardnerella, or like no the known organisms which are or like E. coli. So other than them, these are the organisms which needs to be studied more, like Megasphaeria and Snedia. So these are the strains that we have found out, and the key pathways, which are quite related to preterm, but are proximal tubule bicarbonate reclamation, systemic lupus erythematosus, and then transcription machinery pathways. So these are the different pathways that we have identified. Other than, there are a few other more. I have not written it there here. So there are key biomarkers also, like LEP-A, LEP-X, and RAPOG. These are the different biomarkers that we have identified, and it has been validated using ML techniques and network biology and network making different network studies, and also some statistical methods. So we have done multiple methods to validate that these are the uh, biomarkers that we have identified. So another work that one of our PhD student is doing is it's endometrial receptivity. So endometrium is one of the very important reproductive organ, and endometrial receptivity is one of the very important mechanism in females. There are various hormonal fluctuations that modify the human endometrium, making it receptive and to an implanting blastocyst. So the window of implantation, we call it as WOI, is the receptive condition, and endometrial receptivity uh, causes their loss causes the RIF, that is recurrent implantation failure in women. So this is also in collaboration with National Institute of Reproductive Health and Research in SMR Institute. So they also work on they mostly work on the wet lab part of endometrial receptivity. We have worked on the RNA-seq data of many samples, on huge number of samples we have done. Uh, we have created, we have identified the biomarkers basically. Uh, in endometrial that are important in endometrial receptivity. So differential gene expression, signature genes, parameter selection, parameter scaling, model training, classification models, classified biomarkers. That's all we have done in this project. And ultimately, we have come up with a database which will again soon to be published, which is based on which gives us information about endometrial receptivity and its various factors. So that's all. These are the few glimpses of our work that we have been doing in our lab. And presently, we are mostly working on infectious diseases and mental health because it's a big project. So we are working on methyl-seq data analytics using AI in a very serious mental health uh, condition. So that's all from our side.
Thank you. Thank you so much, Priyanka and Abhishek. That was terrific. In fact, very wonderful works. If you can stop sharing the screen, please, so that we
Yeah.
can move to the big screen. Yeah, thank you so much. Yeah. So, wonderful research over the last 2 decades, Priyanka and Abhi. I was just wondering, just to start with, Priyanka's wonderful presentation, the AI imperative slightly, in fact, by major chance, it differs from generative AI. And in terms of our bioinformatics applications, taking cognizance of these wonderful models that you think for IVF or whatever, how reliable are these particular models with respect to precision. So where are we heading, Priyanka?
So, when I talk about clinical models, we are highly reliant on how well the EHR data has been collected. The major pitfall which happens is that when we are taking this data from a clinical setup, the data is scattered. It is not in the form that we would want it to be for a model. And most of the work goes in the pre-training part. That is, you have to make the data normalized in a manner that it matches your outcome. And in terms of precision, when you talk about, we are still not there 100%. I would not say that even working with data that is non-clinician data, also that we work with omics data, it is easier actually to incorporate, because heterogeneity is one thing that we are dealing with when we are working with the non-clinical omics data. That is one of the problems. But with clinical data, the problem is the data itself is not structured. It is very difficult to collate information that comes from different centers. So that was, the first phase was only with one hospital, but in the second phase, when we started external validation, the main problem started with the input parameters are different for different centers. So when you're talking about reproducibility, I just saw a question, are these reproducible? Yes, if you are working with a certain limited parameters that you're trying to reproduce, it will definitely give you the same outcome as it gave us. But if you talk about that I have a different parameter to incorporate, you will have to retune the data, retune the model, and then use it. It will not work per se for everything. So
Yeah,
no, it will not be precise for everything. Yes, it will work with whatever I have trained it for, the same parameters, it should work.
perfect. Yeah, thank you. It was indeed a very, nice, point. So, I think at some point of time, the AI imperative, needs a kind of a strategic, heterogeneous data sets at a certainty, and then, probably as you
Yes.
rightly pointed out, precision, or even for a better, recall based approaches, taking a good machine learning, heuristics would really, come at a later point of time. I think we have a long way to go, but as you rightly point out, maybe we might need, more amount of those particular, data sets. Uh, something what, what you call, I personally feel is in India, IVF strategy is going leaps and bounds. In fact, many people are opting for IVF, and yet, we are so diverse, we are so, heterogeneous, right from north to south and northeast and, west and all. Even if we are to collect some 10,000 and odd data sets, that are publicly available across the IVF data sets, you know, taking up a better prediction, what is the percent chance that, the IVF results in a positive, outcome, taking some kind of, key attributes that we are taking up is still a kind of a challenge, isn't it?
Oh,
So,
yes,
so,
of
so,
course,
so
I
what
agree.
is your comment on this, Priyanka, or maybe Abhishek? Yeah, please. Yeah.
Yeah, definitely. It's definitely a challenge. Like no, we have not even covered, uh, like no, a very minuscule part of it as compared to the, like no, the amount of IVF procedures that is being happening at this point of time in India. Like no, with the rising situation of infertility. So definitely, I would say that it is definitely it is imperative to consider different like no location or geographical location data sets, heterogeneous data sets to come to certain type of conclusions or model decisions.
Right. In fact, I was reading an abstract somewhere last week. The authors used sharp and line analysis,
Yes.
and they reached consensus. And of course, they couldn't still, get a kind of a good precision on saying that whether or not IVF, could probably give, good results. And in case of India, not only the large amount of data sets, but even the epigenetics play a very important role. On maybe the
Yeah.
diet and all those kind of things, play a very
So, in
important role.
fact, I would say that as an extension of this work, that is IVF, we at in my lab in Amity, we are integrating multi-omics data to these clinical parameters. So now we are coming up with a sort of uh, like a full platform where we are integrating multi-omics
The
data
Sciversation one, I'm located.
integration with this AI model that we
Are there
have
in 15
created.
minutes, Agal,
And
please. Yes.
also we have integrated the large language models to integrate more data into it.
Right.
So it is going to be a wholesome thing that we are coming up in
Excellent.
in future, in probably in another 1 year we are coming up with that.
Right. And what I appreciate largely with these wonderful scientist couple is especially Abhishek, yeah, he is so modest that, he himself has developed a vertigo database. He himself, being, with this
Yeah,
autoimmune condition. And
and that is my
in touch.
passion. Basically, I wanted to dedicate it to my family because my family has this history, and I wanted to dedicate it to my family,
Yeah.
whatever I'm working on, like
Wonderful.
on vitiligo.
So
Yeah.
this shows the wonderful autoimmune thing, how he has translated that particular thing. That shows, what do you call, leaps and bounds of his, praiseworthiness, towards the research that he does. I think we all should applaud, Abhishek on these particular efforts. And of course, how, Priyanka, supports these kind of endeavors. But I was just a bit intrigued. At a certainty, I mean, as a layman who doesn't know about vitiligo and maybe albinism, I know albinism starts from childhood, but vitiligo comes at a later point of time. But are there any common denominators in terms of any biomarkers that we can try to target?
Uh, Like no, yeah, but right. Vitiligo has like no various kinds of. There are different types of vitiligo that we have, and every type has their own pathogenesis. And like for example, we have vitiligo and albinism. Basically, what is your question is that vitiligo and albinism share like no any commonality
Right,
between it. So yes, they share some disrupted, I would say, commonality like melanocytes and or melanin pathways, which but they do not have definitive common clinical biomarkers.
right, right. That's a good
So,
point.
but in India, there is a very big misconception with albinism and
Correct. I was
the
about to,
commonality between albinism
yeah,
and vitiligo. And also, there is another commonality between that people call it as white leprosy,
right, right,
right? So, which is a very big taboo in Indian population, and vitiligo. I personally feel vitiligo is not just a condition. The melanin going off from your skin is a different issue altogether, but it's more of a mental
mental, exactly, yeah.
mental condition,
That's
right? So,
right. Yeah.
so that's
Yeah.
what. So
I mean, vitiligo is an autoimmune, acquired
yeah,
autoimmune condition. It is not something, but albinism is something that you're
yeah.
generally born with, and it
Right,
is not autoimmune.
right,
It is something
But
which is,
Priyanka, there are a few genes which are also common between the in
of course,
melanogenesis. Yeah, I mean melanogenesis, there are some common genes which are shared between
even with
vitiligo.
white leprosy, that was what, Pushpendra Singh was working on, earlier.
Yeah, yeah, white
yeah.
leprosy. Yeah,
But that's a great point,
Yeah.
Abhishek and Priyanka. One or two more questions from my side. In fact,
yeah.
you have extensively worked on vaginal microbiome, and of course, SLE is again this autoimmune disorder. At a certainty, you find large amount of SLE patients, from coming from gynecology and obstetrics. Uh, but at a later point of time, SLE usually develops at a later point of time. Maybe it might even be preceded with gestational diabetes and other things. So again, using explainable AI or AI imperative, can we come up with some models, given these particular data sets that are available? I know,
So
I'm sure you must be working with
I
clinical.
would I would like to add here that we have got a copyright granted for a business model where we are going to it's called micro, it's in my on my LinkedIn, micro pred vedi micro so
Veggie.
basically
Right.
yeah, micro, yeah, vaginal vag micro pred vedi. So basically, this is what this does is like we are going to take samples from from patients, and we are going to do the vaginal
Right.
microbiome analysis using all these techniques that we have established, and we will give proper solution like genetic counselors, like no, so there will be
Right.
like uh, uh, counselors who will give solutions to them, and so this is what was the model, business model that we I have proposed, and I got it copyrighted. Probably in future, if I get a chance, if I get some funders, so I can
Excellent.
start it.
Yeah. Excellent. Thank you so much, Abhishek. We'll quickly have some questions from audience. Abu Peter, please be very quick and brief. Yeah. Abu Peter, yeah, please be very quick and brief.
Thank
Yeah.
you, Priyanka and Abia. That is a very scintillating session, and I am enthused by the presentation. My question is about project 2 presented by Priyanka on sperm infertility, like male-related infertility. During my PhD work, I did some work on male infertility, and I want to ask, did your model put factors like the epigenetic aspect of the Y chromosome aberration? And again, available data set, does it have markers recorded for epigenetic cause of male infertility?
Okay, so
We have
yeah.
we
So
have not.
the
Uh,
no, epigenetic was not part of the study because the main focus was talking about the AZFA B and C prevalence first, and then what is the idea that whether it will be able, you will be able to have a sperm retrieval in these cases or not. So currently, it only talks about locus, that is the physical position of the gene. It talks about the STS or the unique DNA sequence where it has been amplified, and only about the DYS system, that is the prefix, that is it is only talking about these alone standalone genetic markers right now, not the epigenetic thing. And though it is very interesting and the next phase of the work can be done, but since the data sets did not include anything related to epigenetic, because there are many other factors, lifestyle also plays a very important role in terms of infertility. It is not just epigenetic, it is also how you are, because smoking and other kinds of things also lead to these kind of things. So this is also something that we would want to include in future, but right now it was an auxiliary to the PCR kit that is available in the market. So it was designed as an auxiliary product which will be able to help with what is available as the PCR outcome. So right now it is only limited to that kind of thing, which is it is actually very good, because if you have a software integrated along with the PCR technique, it will be helpful for the clinicians in a long way. So
So
that
I would
is
say that my university is after my life to extend it to some other level. So for example, fertility predictor part 2, version 2. So we already have the patent for this. So probably with your suggestion, or probably you may
we can.
collaborate with us, we can come up with
Yeah, I can
with the
see
new
even
version of
Agu Peter has
fertility.
developed wonderful mice models, as a part of his
Yes,
reproductive health things. So maybe he'll
yes,
stay in touch. But that was a
yes.
wonderful question as well, and a wonderful answer. Any quick question, please, Kausar? We are short of time. Very quick, please. I'm really sorry for this. Yeah, yeah.
Yes, thank you so much, Prashant. I'll always try my best to attend your all the shows, so I did today as well. So I don't have any question actually. I follow Abhishek as well. I met Priyanka as well in the last in weeks actually, and you are there in Jaipur. So what I'm thinking, I can correlate one thing very strongly, because we all know, like, this Abir Services Foundation is dedicated for women health awareness and all, and both of them are, like, inclined toward that, how this overall going to improve the health. So can we do one thing? Maybe we can set up another online meeting, and then we can see what we can do in collaboration actually. Because GCCI, I think Abhishek is aware about.
Yeah.
We did wonderful workshop this time, pre-conference workshop in INVIX, and we trained 25 genetic counselors actually across the country.
Yes.
So maybe we can think of how we are going to, connect all the dots that we are here today. Actually, Priyanka is here, you Abhishek is here, and Prashant is here. So we are doing our little bit to connect the all the scientists across the country. But can we formulate something like, even if you can translate into the product at research, that I'm interested in actually.
Sure.
And you rightly said you need some sponsor. I already have some sponsor. If you want, you can translate right away in this month itself. So
Sure.
if you have a great idea, great product, let us bring to the society. Why we have to wait for that?
Yeah.
So
The
that we
purpose
can
of this
connect.
is,
You are very close to me, and I also like to inform you that Dr. Professor BC Das.
Yeah, BC Das.
He was my
Yeah.
director when I was doing my PhD. So on and off, I used to visit the campus a lot actually. But Prashant, next time I'm going to ensure that I'm going to meet Abhishek as well.
very much. I think we should
Sure.
organize a
So
So
workshop
let us,
Dr.
together as well.
Bisidas knows me very well because he's
yes,
also he has started the
yes.
that the course on
ART.
Hmm.
ART, that is
Yeah, yeah, yeah.
assisted reproductive technology course here,
Yes,
and
yes.
and clinical embryology
Yes. Yeah.
course
Yeah.
also.
I'm aware of that project actually, and this
So
wonderful course is going on. And
so
one more thing, Priyanka, you will be surprised to know that one of the project that I completed, before COVID,
Mhm.
related to male infertility, where I screened like a couple of couples actually, and HPV was like very common in infertile male actually, and
Okay.
I personally reported more than 18% HPV infection in semen actually. So that is
Okay.
one of the cause. Even if there is a high chance that recurrent pregnancy loss was there in those HPV infected cases, and even if there was implantation, there is a high chance of failure in future also. So there is a
Yeah. Yeah.
high chance that
Kausil,
is
I'm
not
sorry. I
going
need
to
to stop you here. Kausil, I'm
fertilize the
sorry.
egg. So
You know,
what I'm trying to
introduce
say
and
is those product is already into the market out of that research actually, in the name of Fadsure, and we try to develop into the kit for the male infertility as well as normal screening as well. So let us
Mhm.
do some research where we can not only just to publish the paper. Let's see if something come out for the benefit of the community as well. That I always try to give some more emphasis actually.
Definitely.
yeah,
Yeah.
such essays are there. These essays are definitely many companies have launched such essays
Yeah.
which are related to male infertility. And uh, what like no, we wanted to like no what like no as per the suggestion of Peter, like no, so we can include the other
Factors.
factors
Yes.
into it, and we can definitely come up with a like no much better
So we are going to
comprehensive.
target both the couple actually, male
Yes,
and female. And
yes.
let's say if we come up with something, I think I'm very thankful to Prashant that he allowed me to spare some time and discuss something like this on this forum as
Sure,
well. Thank you,
sure.
Prashant. Over to you.
Yeah, thank you. I'm really sorry. Two things,
Yeah.
because Google Meet and YouTube recording is only for 45 minutes. I'm afraid for that. And second thing, मुझे खाना नहीं मिलेगा because I need to drive 2 hours.
Oh,
So I'm just expecting this to be finished on time. But I think what a terrific civilization it was. I think everyone please switch on your camera, and there is a good photographer sitting in Jaipur. So please switch on your camera, everyone, please, with a lovely couple, and with a wonderful photographer, and a good time here, a great discussion that we have had today. And I think the recording will be live. There will be more discussions. Please ask questions in WhatsApp group. I'm no longer in WhatsApp. I'm enjoying my no WhatsApp days. So yeah, say smile, say cheese. Yeah, everyone.
yeah.
Muskan, Uzma, Regina, Shristi, um, Ellie, Likita. Yeah, if you can switch on, otherwise you'll be off. I'm sorry. Yeah. Say cheese. Yeah, thank you.
Prashant. Thank you so much.
Thank you so
Thank you
much.
so much.
Thank
Let's
you.
connect
Thank
with
you.
our
Thank you
next
so
student
much.
Thank
later.
you for both the
Thank you.
speakers, Abhishek as well as Priyanka. Thank you so much.
Thank you, Abhi. Thank
Thank
you so
you.
much. Thank you,
Thank
Priyanka.
you.
Thank you so much.
And
Thank
Happy New
you.
Year.
And happy
Happy New
Christmas and
Year.
Same to
Happy
you,
New
same
Happy
Year.
New
to
Year,
all of you. Thank you.
everyone. Yeah. Bye bye.
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 Bye.